With that, we move to the next session. So I am excited to introduce Mr. Vishwanth, who is a content writer in a Scioverleaf, and he's going to will be sharing insights on the topic antimicrobial resistance, a bioinformatic approach. Yes, Vishwanth, you can start.
Good evening, everyone. Hope all of you are doing good. My name is Vishwanth, and today I'll be presenting on a critical topic antimicrobial resistance, a bioinformatics approach. So, let's break down this topic into simple terms. So, what is antimicrobial resistance? So, it is when germs like bacteria stop responding to medicines, which makes infections harder to treat, right? So, just imagine a simple infection becoming life threatening because the medicines no longer work. So, in this presentation, we'll be exploring the mechanisms behind AMR, the challenges posed by the traditional approaches towards AMR, and the transformative role of bioinformatics in combating this threat. So, I encourage you to think about the new innovations in science and technology can change the way we approach global health crisis like AMR. My name is Vishwant. Basically, I'm doing my final year in B.Tech Biotechnology at Rajalakshmi Engineering College. I've been a writer at Scioverleaf for the past year. So, getting into introduction to AMR. So, let's start with the basics. So, what is antimicrobial resistance? So, when you get sick, doctor gives antibiotics to kill the germs causing the illness. But sometimes germs adapt and become stronger. So, the antibiotics stop working. So, this is called antimicrobial resistance. Let's begin by understanding what AMR is and why it's such a critical issue. So, AMR occurs when microorganisms such as bacteria, virus, fungi, or parasites develop the ability to resist the effects of medications designed to kill them. So, this means that infection caused by these microorganisms becomes harder to treat, which leads to a prolonged illness as well as higher medical cost and increased mortalities in serious conditions too. So now considering by 2050, this AMR is projected to cause around 10 million deaths annually. So it is a figure which is higher than the deaths caused by the cancer today. Just imagine the strain this would put on healthcare systems, especially in developing countries where resources are already limited. So, what causes AMR? So, we know that we just looked into what is antimicrobial resistance. So, what may cause it? Even though it is a very rare, a very rare thing which microorganism undergo through naturally, there are some artificial ways in which antimicrobial resistance is developed in microorganisms. So uh, we can actually relate it to everyday activities also. So the first one is overuse of antibiotics. So when you get sick, you take antibiotics, but sometimes in case of very simple cold, common cold, we take a lot of antibiotics. So what happens is it creates a lot of stress on our body, and the microorganisms develop a resistance to these antibiotics. As well as in some cases in agricultural use, antibiotics are used, which introduces these resistant germs into our food and environment. For example, in poultry chickens, in order to get the desired size and weight within a period of time, antibiotics are just loaded into the chicken. So this is also a reason for this antimicrobial resistance. And lack of hygiene is also a reason for AMR because poor infection control in hospitals and communities allows resistant germs to spread. So understanding AMR isn't just a medical issue; it's a social and economic challenges too. So we just looked into what is AMR and how like AMR is promoted. So let's look into the mechanisms of AMR. So how this AMR is developed by the microorganisms. So let's think of germs as a clever escape artist. Like have you seen in the you must have seen in the movies like Prison Break. The main characters will take a lot of ability to just they will use the tricks to move out of the jail. Okay, so they in the same way the germs also use tricks to survive antibiotics. So Microorganisms have evolved sophisticated mechanisms to evade the effects of the antimicrobial agents. So let's delve into four, generally classified primary mechanisms. So mutations in target sites. So it is similar like changing the locks. Germs can change the part of themselves that antibiotics target, so the medicine no longer fits. So what does the medicine do? So the medicine just targets a part of the microorganism and disrupts it so that it may damage the function or structure of the microorganism so that it can no longer live or reproduce. So what happens is when antibiotics target specific bacterial components, mutation can alter those components, preventing effective drug binding. Just think of antibiotics as arrows and bacteria as targets. If the targets keeps changing, the arrows can't hit them anymore. The second mechanism is efflux pumps. So just it is like throwing medicine out. So some germs or some microorganisms have pumps to push antibiotics out before they can harm them. So these are like the molecular pumps that bacteria use to eject antibiotics from their cells. So this reduces the drug's concentration inside the cell, rendering it ineffective and allowing the bacteria to survive at a higher antibiotic concentrations. So third one is enzymatic inactivation. So it's similar like breaking the medicine. So certain microorganisms releases enzymes that destroy the antibiotics. So some bacteria produce enzymes such as beta-lactamase, which break down antibiotics before they can act. Actually, this is a major problem with drugs like penicillin. So we already know that penicillin is antibiotic which acts against gram-positive bacteria. So what does this penicillin does is it prevents the formation of peptidoglycan, which is a very important part of the cell wall of the gram-positive bacteria. So the microorganism just develop the resistance to these antibiotics by producing penicillinase. It is an enzyme which is used to break down penicillin so that it can no longer work on them. So the final one is the horizontal genes transfer. It is sharing the tricks. Sharing, as I discussed before, they are tricksters. Like they use tricks to evade from the antibiotics. So what happens is the microorganisms can teach other microorganisms to resist antibiotics by passing on their resistant genes. So this allows bacteria to share resistant genes with one another through processes like conjugation, transformation, and transduction. Okay. This conjugation, transformation, and transaction are methods by which genetic materials transfer from one bacteria to other. It may be through direct contact or other mechanisms too. So this accelerates the spread of resistance within the bacterial populations. So the traditional approaches towards this antimicrobial resistance has a lot of limitations. So traditional methods for identifying and this combating AMR have significant drawbacks. The first one is it is highly time consuming. So the results can take days. It may delay the treatment. For example, in culture-based techniques, it can take days or weeks to identify this resistant strains and delaying the clinical treatment. And it has a very limited scope too. So it misses new tricks. Since it is a traditional approach, it is not quite updated. Okay. So some resistant mechanisms are hard to detect. So traditional tests might not detect new or less common resistant mechanisms and higher cost. So advanced testing methods are often, unaffordable in low-income regions where this antimicrobial resistance hits the hardest. So these limitations highlight the need for the advanced tools that are faster, more accurate, and more accessible, which brings us to the role of the bioinformatics. So bioinformatics in antimicrobial research. So let's look into what is bioinformatics. So bioinformatics is like giving scientists superpowers. So what happens? What is bioinformatics? Bioinformatics integrates biology, computer science, and information technology to analyze biological data. So its applications in AMR are transformative. So the first one is data analysis. It is used to find the patterns. So by processing vast data sets, bioinformatic tools can identify resistant patterns and predict the future trends of the microorganisms. For example, by analyzing hospital data, bioinformatics can spot outbreaks of the resistant germs early. Okay. Then the second one is the genome sequencing. So what is genome sequencing? It is nothing but just studying the microorganism's genetic material, that is DNA. So just like humans have DNA, microorganisms have to their own genetic material. It may be DNA or RNA. So by reading their DNA, we can identify the genes making them resistant. So the whole genomic sequencing identify resistant genes and provides insights into their mechanisms. This helps in predicting how resistance might spread in the future. And the third one is the drug discovery. It is nothing but just discovering new medicines. So computers can quickly analyze millions of compounds to find potential antibiotics, something that would take years using traditional methods. So to discover a drug, it is a very tiresome process and it takes a lot of time because it includes a lot of preclinical testing, clinical testing, clinical trials, post-clinical trials, and after that, after 15 to 20 years only, the drug can enter into the market. So this machine learning algorithms can analyze data to predict the antimicrobial resistance and identify the potential drug candidates. These insights are invaluable for pharmaceutical companies developing new antibiotics. So genome surveillance and the data sharing. So why are we looking into this topic now? So one way bioinformatics helps is by tracking the spread of the resistant genes worldwide. Okay. So large scale genomic projects plays a crucial role in monitoring these AMR trends. The Global AMR Surveillance System, GLASS. Okay. It is established by the World Health Organization. So what it does is it integrates data from thousands of laboratories around the world. It uses the bioinformatic pipelines to analyze the genomic data from the bacteria, so detecting emerging resistant patterns and identifying novel resistant mechanisms. Okay. So data sharing platforms like a global microbial identifier GMI are also critical in advancing AMR research, allowing researchers to pull their genomic data and compare resistant patterns across different regions. Okay. So before we move on to the next topic, I want to make sure that you are getting to know about what are the basics of bioinformatics. So a database. So database, it is like a collection of data. So database is a server. Okay, it is a web server which contains all the information related to the gene or protein or specific information which is like specific to that database. For example, NCBI, National Center of Biotechnological Information, is a government-based database which has information related to the genes of the all organisms. So what happens? So what do bioinformaticians do is they just retrieve the data and they just analyze the data and they just sequence, predict, or they just comprehend the data and they just obtain the results. Okay. So in this genomic surveillance, let's look into a real example. So during the COVID-19, scientists used genetic tools to track how the virus was changing. So similarly, bioinformatics can track resistant germs to stop their spread. So this programs like the World Health Organization's global AMR surveillance system collects and analyze the microorganism data worldwide. It's like having a global weather map, but for microorganisms. So the next one is whole genomic sequencing and the antimicrobial research and analysis. So, what is whole genomic sequencing? You know what is sequencing, right? Just comparing, two sequences and finding the similarities and dissimilarities and aligning them, and like predicting their alignment scores as well as predicting their common ancestors, and this comparing their functions. So lots of stuffs are there in analysis. Like this sequencing. So what is whole genomic sequencing? So bioinformatics who uses whole genomic sequencing to read germ DNA, that is the microorganism DNA, like a book. It is like like taking the entire gene of the microorganism and just putting it in a book and just reading the whole book to analyze it and getting the information. Okay. So this whole genomic sequencing provides a comprehensive view of a microorganism's genetic material. So here's how it works. The first step is sequencing. So the whole genomic sequencing deciphers the entire DNA sequence of the bacterial isolate. So the scientists read the entire DNA of a microorganism like reading every page of a book. The second one is annotation. So bioinformatic tools find specific words or genes which are responsible for the resistance. So they just annotate it. Annotate is nothing but marking in simple terms. So third one is analysis. So researchers use this particular data to pinpoint the resistant mechanisms and explore potential drug targets. So it shows how germs spread between people, animals, or even the environment. This information is used to develop better treatments and stop resistance from spreading. For example, in a particular study, researchers used this whole genomic sequencing to find out why an antibiotic wasn't working on E. coli. They discovered that a specific resistant gene which helped doctors choose the right treatment. Okay. So the next one is metagenomic analysis. Okay. So what is metagenomic analysis? In simple terms, let's look into it. So sometimes instead of studying one germ, scientists look at an entire microbial community in places like soil, water, or the human gut. This is called metagenomics. It's nothing but studying the gene of a of a complete microbial community in a particular environment. So why is it important? It helps us track resistant genes in the environment, showing how farming or wastewater can contribute to the antimicrobial resistance. So it supports the One Health approach, which is nothing but it connects human, animal, and environmental health. For example, scientists took samples from rivers near farms and found that resistant germs in water. Okay, so this showed that how farming practices were contributing to the AMR. As I discussed before, in the poultry farms, like when you abuse when you overfeed the chickens with these antibiotics, it is also a background factor for this increase in the like antimicrobial resistance. Okay. So what are the bioinformatics tools which are required for this antimicrobial research? So some widely used bioinformatic tools in antimicrobial research includes a BLAST. So what is BLAST? It is nothing but basic local alignment search tool. So it's nothing but just aligning two sequences of different organisms or closely related organisms, and we will study the similarities of the amino acids or of the gene. So we will be studying its function, expression, and by comparing these two sequences, we can get a lot of information on its ancestors, as well as a lot of stuff can be interpreted from these sequences. Okay, so it is a tool for comparing an input sequence against a database to identify genes associated with resistance. So it compares the the microorganisms DNA to a database to find resistant genes. So these resistant genes are already, scientifically proven, and they're uploaded in the database. So when you search for a particular organism, the resistant genes are already there in the database. So when you compare them and align them by uploading these, raw sequences into the database, it will just give you all the resistant genes of that microorganisms. Okay. So this BLAST is a fundamental in detecting mutation linked to the antimicrobial resistance. The second one is REST finder. So what is this REST finder? So this tools identify resistant genes in bacterial genome by comparing these genomic sequences to a database of known resistant genes. So in basic terms, what does it do? It detects resistant genes in the the germ DNA quickly. So it's widely used for surveillance and epidemiology. Okay. So nothing but it identifies resistant genes in bacterial genomes. So the next one is AMR++. So it is also a software tool that uses the same sequencing data technique to detect antimicrobial resistant genes and mutations. So it is also particularly helpful in clinical diagnostics, offering a fast real-time analysis of pathogen genomes. So fourth one is Proca. So Proca is a tool which is used to annotate bacterial genomes. Mark the bacterial genomes at particular positions. So identifying resistant genes and other relevant features. So Proca is essential for understanding the genetic basis of AMR. Okay, and the deep ARG. So it is a type of a deep learning based tool. It is a what is deep learning? We will be looking into the next slide. So this deep learning based tool predicts antimicrobial, antibiotic resistant genes from genomic data. So this leverages the machine learning algorithm, which improves the accuracy and efficiency to a very vast amount. So these tools have streamlined this antimicrobial resistant research by automating processes that would otherwise be time intensive and labor intensive too. So artificial intelligence and deep learning for predicting resistant mechanisms. So, what is artificial intelligence? So, what is the most used artificial intelligence in our day-to-day life is we can think of this Chat GPT and Gemini, and many AIs have become a very vital part of our livelihood, right? So, right from writing the assignments or finding any solutions, we just approach this AIs. So this artificial intelligence and machine learning are the very great game changers in the AMR research. So what is the artificial intelligence? It is a set of technologies that enable computers, to perform a variety of advanced functions. So it includes the ability to see, understand, and translate spoken and written language, analyze data, make recommendations, etc. Okay. So, what is the role of this AI in AMR research? So, predicting resistant profiles. So, the artificial intelligence can analyze genomic and clinical data to predict bacterial resistance, aiding in personalized medicine. So, this word personalized medicine, it is blooming as days goes by. What is personalized medicine? So, according to a patient, and just concentrating entirely towards that particular patient's genome, and taking his or her genome and just analyzing it and just like giving what he or she exactly needs is personalized medicines. Okay. And the second one is identifying novel mechanisms. So deep learning is a type of artificial intelligence. So deep learning is also a type of artificial intelligence that uses artificial neural networks. So what is artificial neural networks? It's nothing but it's like it mimics the neuron, like in our brain, in our nerves. So what happens in a neuron? So the signal travels from the dendrite to axon. So in the same way, it is like the data and the input and the output are just represented in the form of a neuron. So why does our neuron is like very highly efficient? Because the signal just travels in milliseconds, so it is a very fast mechanism. So the same way, the same technique is used in this deep learning techniques. Okay, so this uses the artificial neural networks to teach computers how to learn data, learn from the data. So deep learning identify previously unknown resistant mechanisms, guiding new research directions. So this AI is trained based on the training data and testing data. So what is the training data? You'll be providing a particular data and will allow that the machine learning algorithm to just train on the data set, and after that it is used. So this particular data is used to predict, like unpredicted data. Okay, so accelerating that discovery is also an application of AI. So this AI screens large data sets to identify potential antibiotic faster than its traditional methods. Okay, so one of the best example is the discovery of a new antimicrobial compounds using AI models trained on chemical databases. So computational modeling and simulations. So why it helps in the predicting the spread of the resistance. So this computational modeling, it is nothing. It is also called in silico modeling. So what is in silico modeling? So the use of computers to simulate and study complex systems using mathematics, physics, and computer science is called in silico studies. So in silico studies is completely based on theoretical part. Okay, it uses maths, physics, and computer science. So computational models, which are used to simulate the bacterial growth, resistant evolution, and treatment scenarios. For instance, these models can predict how bacteria might develop resistance to a new antibiotic, or helping researchers optimizing its use. So these simulations are also used to design strategies for minimizing, the development of resistance in hospital settings. So, integrating multiomic data, it is a comprehensive view of AMR dynamics. So, what is multiomics? We see that integrating multiomic data. So what is multiomics? Multiomics in simple terms is nothing but it is a biological analysis method. So it is an analysis method that combines multiple omics data sets to study biological system. So what is omics? We see in that it combines multiple omics databases. Okay. So what is omics? Omics refers to the study of the biological molecules such as genes, proteins, and metabolites. So integrating multiomic data offers a comprehensive view of the resistant dynamics. So what is genomics? Genomics is nothing but it reveals the genetic makeup of the bacteria, nothing but the DNA, RNA, all the stuff. Okay. The second one is the transcriptomics. So what is transcriptomics? It identifies the active genes contributing to the resistance. It is nothing but the mRNA, messenger RNA. So it is like a central dogma theory. So from the DNA, the mRNA is synthesized. Okay. So the study of the entire mRNA is called the transcriptomics. The third one is the proteomics. So what is proteomics? It is a study of the proteins that are involved in the resistant mechanisms. And after that, what happens is metabolomics, the study. This study explores metabolic pathways of the bacteria that supports the resistance. This entire, the sequence is just like a central dogma theory from the gene DNA to mRNA, mRNA to protein, and protein to protein expression. So protein is involved in a lot of pathways, metabolic pathways. So these pathways is explored, like to find the antimicrobial resistance. So combining these data sets provides a holistic understanding of AMR, guiding research and intervention efforts. So let's look into real life case studies, of this antimicrobial resistance. You know that Mycobacterium tuberculosis is is responsible for causing a deadly disease called tuberculosis. It is a respiratory disease which is it has high mortality. It is a like a very deadly disease. So it is very notorious for its ability to develop resistance to multiple antibiotics too, including the first line drug rifampicin. So this mycobacterium tuberculosis is very well known for developing resistances. The first line drug rifampicin is the first drug which was used to treat this tuberculosis, but it is well known for developing resistance to all sort of antibiotics. So recent efforts to combat this drug resistance TB. So this TB is called drug resistant TB because it is resisting to the drugs. It has been focusing on using bioinformatics to map the mutations in the RPOB gene. So RPOB gene is responsible for causing this resistance in the bacteria. Okay, so which confers that the rifampicin. So how does exactly this rifampicin work? So it binds to the bacterial RNA polymerase and inhibiting its ability to transcribe the DNA. So from DNA to mRNA, this one, like the rifampicin just halts the process at that particular step. So this microorganism cannot function. Okay. So high throughput sequencing technologies. What is high throughput sequencing technologies is just sequencing technology which can analyze a large amount of data in a shorter period of time. Okay. So this is combined with the bioinformatic tools like TB profiler and microbe, which have allowed researchers to rapidly identify this drug resistant strains, providing faster and more accurate diagnostics, which is very vital for the effective treatment. So the tools like the TB profiler map mutations, conferring this rifampicin resistance. Okay, so the rapid diagnostics have significantly improved the treatment outcome for this drug resistance TB. So till now, we have seen what is AMR, how this AMR works, what is the mechanisms, and what are the different approaches to antimicrobial resistance, and what are the applications of bioinformatics in antimicrobial resistance. So there are lots of just like traditional approaches. These bioinformatics also have its own challenges and limitations. So while bioinformatics offers powerful tools, there are challenges and limitations too. So the first one is the data quality and availability. So the reliable, high quality genomic data is very crucial for accurate analysis. But however, in many regions, especially low and middle income countries, this access to sequencing technology and data sharing platforms is very limited. Okay, so the next one is the complexity of the resistant mechanisms. So the microorganisms can resist in ways we don't fully understand yet. It's still growing. It's still growing. It's still adapting. It's still resisting it. It's a very crucial topic which we need to understand day to day life. Okay, so this antimicrobial resistance is influenced by multiple factors, including these genetic mutations, environmental factors, and horizontal gene transfer, which I already discussed, making it very difficult to model and predict accurately. The final disadvantage is ethical and privacy concerns. The use of the genomic data it also raises ethical issues, especially with regard to patient privacy and concern for data sharing. When you use a patient data, all the patients will not be willing to share the person, their personal gene data, to for further studies. So it raises ethical issues. So using this genetic data raises questions about privacy and consent. So, concluding this antimicrobial resistance using bioinformatics, in conclusion, this bioinformatics is reshaping AMR research to a greater extent. So, to tackle this, global issue, it is required, this global health crisis, and to win this fight against AMR, we need, like increased data sharing and collaboration. This increased data sharing, among researchers is very crucial, for accelerating AMR research, and which will speed up the finding effective solutions too, and continued advancements in AI and machine learning. And like this advancements it is already occurring, but we have to focus more on this antimicrobial resistance. So this continued advancements revolutionize this AMR, so it leads to a lot of new insights, therapies, and better treatments. So third one is public awareness. So raising public awareness of this AMRs is very essential for fostering this antibiotic use and promoting a collective effort to combat this threat. So like a lot of people do this stuff, simple stuff. Okay, So when a doctor prescribes you antibiotic for your particular sickness, most of the what most of the patients do, that's the basic mindset. They take the medicines, antibiotics for 2 days. If it is prescribed for 5 days, what happens is once you take the antibiotics continuously for 2 days, the illness may reduce to a extent. So what happens is the patient just things that okay, the microorganism is dead inside my body, so I no longer need to take antibiotics. So what happens is the microorganisms, they are not 100% they are not dead. Okay, many organisms become dormant. So in the meantime, they will be developing alternate strategies to overcome these antibiotics. So what happens is you again develop the disease. So even after developing once again the disease, even though you take the antibiotics, it no longer works on that particular microorganism because it has already gained resistance against it. And this AMR is a very serious challenge, but bioinformatics offers hope. So it helps us understand how germs resist antibiotics, and it guides the development of new treatments. It supports smarter, faster responses to outbreaks. So by combining scientific innovation with global cooperation, we can build a future where AMR no longer possesses a major threat. So by concluding this, I thank you for your attention, and I am happy to answer any questions.